can you tell us a basic question? Is it Bilophila a friend or a foe?
So if you put it in a scale, it would balance towards the bad side.
It's like kind of making small breweries inside the gut.
Exactly.
Hello Liz, thank you for joining us today.
Hi Yasir, thank you for the invitation.
You have been working on Bilophila for a long time, about a decade. And before we deep dive into your research, can you tell us a basic question? Is it Bilophila a friend or a foe?
It's a common bacteria from the microbiome, but it's now being recognized as a bacterium that potentially is bad for us. So if you put it in a scale, it would balance towards the bad side.
So what made you work on Bilophila?
Well, my background is in the sulfur metabolism. If you're familiar with sulfide, this is a molecule that is, it smells like rotten eggs. So it's widespread, and if you go to a wastewater treatment plant, you will be able to recognize it just because of the smell.
Oh, okay. So that's hydrogen sulfide gas, the bad egg-like smell that comes from Bilophila.
Yes, it's a byproduct of their metabolism.
Nice. So what kind of research questions you have been studying in the last few years?
So my main driver has been host-microbe interactions. So in this case, I wanted to know how Bilophila colonizes the host. That was the main driver of this study. And for this, we use a variety of techniques, including mutagenesis with a technique called TRADIS. We also use metabolomics to know which metabolites were there, and transcriptomics to know which genes were being expressed by the bacteria.
Oh, that's interesting. You have been using cutting edge techniques. But like, what was your experimental setup? How you did the research basically? What questions you asked?
Yes. So the main question was, how do they colonize their host, and what's their impact on the host when they are by themselves or with a community itself? We have the advantage that we have access to germ-free mice facilities, so we were using these germ-free mice to colonize the mice groups with different groups of bacteria.
So if I understand it right, you used germ-free mice and you used 3 different communities. In the one community, you infected with Bilophila only. To the other community with the Sihumai, that is your microbiome. And the third one, Bilophila plus that good bacteria or the Sihumai you were using.
Exactly.
Okay, so that's very interesting. So Sihumai could be control, and Bilophila we know is a bad. So the combination should be somewhere in the middle.
Well, that's what we expected at the beginning, that the impact on the host would be the worst when Bilophila was by itself, mainly because we would expect that the bacterium would be at higher abundance by itself than compared with the community. But it turns out that, in fact, they did have the highest number of Bilophila when it was by itself, but the worst impact on the host was when Bilophila was together with the microbial community.
Oh, wow. That's a bit strange, isn't it? Like, Bilophila on its own couldn't cause that much harm, but Bilophila with good bacteria was causing more harm.
Yeah, well, I should point out, it was not only good bacteria, it was the representation of the whole microbial community.
Oh, so it's a kind of a microbiome, good and neutral bacteria kind of mix.
Yeah.
As you mentioned, you are using quite advanced techniques. What did you find? Which technique did you use to find out what is going on when Bilophila interacts with the microbiome, and why it gets worse?
So we use a technique called TRADIS. In this technique, we create an array of different mutants.
Okay.
This means that different genes will be broken, they will not work, and you pull all this combination of mutants, put it in a specific condition, in this case the mice, and we could basically get the number of mutants that were present in the gut compared to our in vitro conditions, so the flasks.
Okay, so if I understand it right, like you make mutants where every mutant has breakdown at different part of the gene.
Exactly.
In the community.
Yes.
Oh, okay. So, and then you try to understand what's going on.
Exactly.
So it's like, taking parts of the engine of a car one by one out and see that, like, okay, what part is doing what? Is that right?
Exactly, yes.
Okay. So what did you find out then?
So we found that microcompartments were essential for colonizing the gut. And microcompartments are these protein structures that are formed by the bacteria, kind of like organelles, to contain everything that would be toxic as a byproduct from their metabolism. Thanks to these microcompartments, they can use taurine, which is an amino acid that we get from food. It's also common in your energy drinks. And we need this amino acid. And Bilophila uses this amino acid and finally produces as byproducts ethanol, which is an alcohol, hydrogen sulfide, and acetate as byproducts.
Wow, there are quite a few things in there, especially the microcompartment. And as you mentioned, like ethanol, H2S, hydrogen, so it looks like Bilophila is quite a can feed on quite diverse range of metabolites or the starting energy sources.
Yes, one of our hypotheses of why it's producing more ethanol is mainly because it's competing with a lot of resources with all the members of the microbiome that are present. So it's kind of competing for the same energy sources that are there, including hydrogen, including formate, and so on. And because of this competition, they are probably shifting their metabolism to use other energy sources that shift the flux towards producing more ethanol.
Wow, so it's very adaptable.
Yes, it is.
So is it possible that the Bilophila is making other bacteria to produce something that it is using for energy and producing anything harmful for the host? That's why we are seeing the worst effects on the host. Was it making other bacteria to produce ethanol?
Possibly.
Wow, it's like kind of making small breweries inside the gut.
Exactly. So we don't know who would be the main producer of ethanol. We know that Bilophila can do it. We proved it by just having metabolomic analysis of just Bilophila and seeing how it consumed or produced ethanol. But the other members of the microbiome could also potentially produce ethanol as well.
Okay, that's cool. So what kind of effects it has on the host?
So alcohol can travel to the liver through the gut-liver axis and cause a similar effect to what you would have in the non-alcoholic liver fatty disease. In which case, although you hear the word non-alcoholic, in fact, people that are suffering from this disease have increased levels of alcohol in their feces. So it's similar to what we observed in mice. We also observed higher levels of ethanol in the feces of the mice.
What do you think? Where do we go from here with all the knowledge you have got and with these research results?
Well, we first need to investigate how Bilophila responds to different microbial communities. So next, we will be investigating how the microbiome of people that are suffering from obesity relates when Bilophila is present compared to people that have a lean microbiome. Only around 50% of the population have Bilophila, so in the future we could use Bilophila as a marker to know if potentially you could have a worse outcome for the liver or for your levels of cholesterol and so on.
Okay, so you mentioned fat here, or obesity. So does it has any relationship with Bilophila?
Yes, this bacteria thrives with fat. So it loves fat in general.
Okay, so if I eat anything fatty like cheeseburger or something, I should be worried?
Maybe. So depending on the food you are eating, you will get a different microbiome. And in this case, food that is rich in animal protein, milk, red meat, and so on, will make Bilophila bloom.
All right, okay. That's interesting. So, so for our listeners, what is one takeaway message that you would like to give?
Well, the microbiome is just one part of us, but the community and how many of them we have will change depending on what we are eating. So we always have to be mindful of what we are eating.
Okay, so listeners, as you heard from Liz, whatever dietary choices you're making, that really affects you. When you are making a dietary choices, you are not just making for yourself, you are making for the communities inside you. So make some smart choice. Thanks a lot, Liz. It was really lovely talking to you, and thank you for all the insightfulness that you have given us today.
Thank you. It was my pleasure.
Thank you. Until then, this is the Soundbite.